So a really interesting topic we're here to discuss today, the question of where pharmacogenetics and disease stratification can contribute to making safer and more effective drugs, and what's happening in practice and what the challenges are. We've got a great panel of colleagues joining us today to guide us through this topic, and as you've seen from their biographies that have been sent out, really interesting views. And we also know we've got a mix of participants today. Some of you are experts in this and some of you are new to this. So please do ask any questions about, what this topic's about and its application within drug discovery, and we'll get to those. And how we're going to run the next few minutes is I'm going to ask each of the colleagues on the panel to talk through their thoughts, then we'll have a discussion, and then we'd love to hear your questions and comments, either as part of this panel or within the discussion groups later. While the panel are talking, if you want to put questions in the chat as they come to you, please feel free to do so. We'll look at that. And as Munir said, we'll also have an opportunity to raise hands later, so whichever works best for you. So let's get started then. First, I'd like to bring in Ellen McDonagh, who's the Informatics Science Director of Open Targets and the group leader for the Open Targets core team at the European Molecular Biology Laboratory and the European Bioinformatics Institute. So Ellie, over to you.
Thank you, Sheuli. Really excited to be here today. Thank you everyone for joining. Yes, so we're Open Targets. We're a partnership between industry and academia. And we systematically bring together evidence between targets or genes and diseases. And we do this through our experimental program to generate novel data, as well as then our informatics program, which will bring together the novel data that we generate and publicly available data sets. Use some novel techniques and methods to generate new insights into that data. And we also have our Open Targets 2 informatics resources. So Open Targets Genetics, and these are open source, available to everyone. Open source genetics that we bring in large genomics data from UK Biobank, for example, all of the GWAS catalogue, GWAS studies, and combine that with functional genomics data to help predict the most likely causative target underlying GWAS associations. And we also have the Open Targets platform that brings together not only those predictions from Open Targets Genetics, but other data resources such as Genomics England Panel App, ClinGen, to bring together evidence between targets and disease associations and tools within the platform that help you prioritise the targets that you're looking for, or based on the diseases that you're interested in. And on top of that, we provide annotations such as cancer biomarkers, for example, responses to drugs and drug information that help people build their therapeutic hypotheses and further sort of prioritise the best targets for particular disease or subset of diseases. But I'm interested today to find out, sort of scope out how pharmacogenetics is being used across drug discovery, but also in particular in early target discovery and prioritization, and if there are additional data sets or additional data that we should be bringing in, for example, to the platform, and utilizing that to help people prioritize their targets based on pharmacogenetics information. So we do bring in some drug response information, such as pharmacovigilance data from FAIRS, black box warnings, and why clinical trials are terminated. But the challenge that we find with pharmacogenetics data is linking it to a particular target, because often, for example, adverse drug reactions are off-target, have off-target effects. So our challenge is sort of how we utilise pharmacogenetics data to help prioritise targets, inform on safety, etc. of those targets. So I hope that was comprehensive, Sheuli.
Great, thank you, thank you. And we'll pick up on a number of those points in the discussion. And just a reminder to everyone, if you think of questions, do feel free to put them in the chat now if you'd like to. So thank you, Ellie. We'll move on to Dr. Charlotte Green from the University of Dundee Drug Discovery Unit. Charlotte, would you like to share some thoughts?
Hi, thanks, Sheuli. So the Drug Discovery Unit is set within the University of Dundee. We are an academic drug discovery unit run like a biotech company. So 70% of our 130 staff came from ex-pharma. So everything is run sort of to a pharmaceutical standard. We work in anti-infective space mostly. About 70% of our portfolio is in that space, so malaria and connectoplastid diseases, so parasitic diseases. However, we have about 30% of our work focuses on innovative targets. So these are any diseases of unmet medical need and novel drug targets. And part of what we've been doing with the UK Spine is helping to triage and identify novel targets that can be used in healthy aging to treat multimorbidities of aging. So essentially with the focus to put people on less drugs as they age, the drugs that treat multi-diseases. And we've been working closely with Ellen and her colleagues at Open Targets to incorporate data in terms of GWAS studies, pharmacogenetics, and all these things to try and identify better targets to take forward into drug discovery pipeline. So the DDU has the capability to go all the way through the drug discovery pipeline to deliver clinical candidates. So we have our own internal drug metabolism and pharmacogenetics group. And I think this is where we're also trying to use pharmacokinetic data to better our mouse models, for example. How can we map some of these things onto what we do in that space so that things are more likely to make it through the clinic? So things that you've probably heard about humanized mice. So we have our own internal humanized mice there, but how can we keep these things improving? And as Ellen said, it's difficult to identify targets sometimes that are causing these adverse effects and stuff. It's fairly simple with the pharmacokinetic data that we have to identify the metabolites, the metabolic enzymes, the transporters, but wider than that, it is more tricky. And we've been trying to apply this to identifying the best targets at the front end. Yeah, what are the best targets that are going to be less likely to suffer from pharmacokinetic genetic problems? You know, what are the good targets and how can we get around these in a personalized medicine sense? And the more we can understand that, the better we can hopefully make drugs for the general population. Great,
thank you, Charlotte. So our next speaker is Dr. Maria Falaguera, who's a postdoc researcher at EMBL-EBI. Hi, Maria.
Hello, good morning. Thank you for inviting me to this conference. So I come from a chemoinformatics and systems pharmacology field, and I recently joined the Open Targets group. I'm working with Ellen McDonagh in the target identification and prioritization of targets associated to age-related comorbidities mainly. I am very interested also in assessing the level of target novelty in order to discover new drug opportunities in this field. And although my notions about pharmacogenomics are still very vague, I am more and more becoming aware of the implications that genetic diversity across patients with similar diagnosis may play in drug response. And I found particularly relevant this in polypharmacy scenarios where many targets are being, many drugs are targeting different proteins. And I would like to, during this session, explore how could we improve our understanding of how these drug protein networks work. Thank you.
Thank you, Maria. And then on to Dr. Alex Phipps from AstraZeneca.
Thank you, Sheuli. Good morning, everyone. It's a pleasure to be here. So my background is as a biochemist, but I've worked in clinical pharmacology and modeling for many years now in industry. And one of the things I've noticed is how we've progressed in terms of targeted therapies, either using biologics or small molecules. One of the ones that's been a recent success is in the treatment of spinal muscular atrophy, with the knowledge that the SMN1 gene deletion can be overcompensated by upregulating SMN2 through antisense oligonucleotides. So it's really targeted to a single genetic mutation. I work most of my time in oncology, though, and actually as a clinical pharmacologist, we're the recipients of information from the discovery area. And there have been a whole host of different identified genetic mutations which leave patients at predisposition to different cancers. And again, there are medicines that have targeted things like EGFR, ALK, BCR-ABL, where there are patients are at high susceptibility BRCA mutations as well. Sorry for all the acronyms. I can explain them in the chat box after. But it's not only that you block a target, a specific target. You can also have a situation where you can adapt the immune system. So, for example, in colorectal cancer, patients historically didn't respond to checkpoint inhibitors. But it's known that about 10% of patients have a disease called Lynch syndrome, which is characterized by a deficiency in mismatch repair enzymes. And actually, those patients have high immune cell infiltrations within their colonic tumors, and 50% of them respond to pembrolizumab. So it was not a situation where you were blocking a target. It was you were retargeting the immune system based upon the likelihood of response. I think that's pretty neat. So we're looking at all sorts of different ways to try to target better medicines and actually better recruitment of patients. This is really important. Getting patients into trials where they are most likely to respond. The days of all comers in early trials and trying to pick out a signal are going. And I have to say that I think that is progressive. And certainly if it's a sort of grandmother test in terms of would you let your grandmother in a trial, I think this is really important, that we're doing our best to make sure that we're getting the treatment to patients, and then you expand once you see a clinical signal. In terms of clinical pharmacology, though, we're very interested in things like genetics driving combination treatment therapies, drug interactions, of course, that's a big area, but also dose adjustment based upon levels of expression. And one of the classics is with receptor expression or expression differences of CD20 in different leukemias. So, for example, with chronic lymphocytic leukemia, the dose of a drug like rituximab is about twice as high as it is for patients with diffuse large B-cell lymphomas, because their B-cells expression, that CD20, are higher. So we're adapting all the time, and this is really important. And this is what gets me and my team out of bed in the morning, really thinking about how we can use this information that has a pharmacogenetic background to try and make better medicines and better doses for patients as well. With that, over to Sheuli.
Great, thank you. What a great introduction to a topic. Listening to what our panel have said so far, I think we've kind of got two broad sets of things we can explore. So the process of drug discovery and the how and the what and the challenges and the uncertainty. But I think all of our speakers talked about the important bit of actually what does this mean for patients in terms of actually whether that's polypharmacy or adverse events. So over the course of the next while, it'd be good to explore both of those aspects. But I think maybe if we start with we've had a good overview, maybe from each of the panelists, what do you see as the sort of the main challenges? I think some of you have alluded to them, but what when you're thinking about this topic, what are the main challenges for you? Who would like to go first? Alex?
Sure, yeah. How long have I got? There are a number of challenges. I mean, the first one is always causality versus correlation. What you're looking for, is it real or not? And I have to say, in over 20 years in the industry, I've seen a lot of rabbit holes where we've gone and asserted causality when it was actually only correlative. You also don't know, sometimes you can have a pharmacogenetic mutation which is actually downstream to the core effect. So I've seen examples like that as well. But that's basically the best way to approach this is with just an open-minded scientific pursuit and testing the hypothesis as much as possible. I think there's an ethical position as well, and I'm going to go back to the example I gave with pembrolizumab before, the checkpoint inhibitor. Patients can be stratified or treated based upon the level of expression rather than an absolute, is this target expressed or not? And that's really interesting because patients with a immunohistopathology complex level of on a grade of 2+, 3+, or up to greater than 49% of cells expressing PD-1 do better, and the response rates might be 30–40%. But patients with PD-1 expression 0 have a 10% response rate. So what does a physician do? Do you not treat those without expression because they don't have the same likelihood of response as the higher? Or do you say, well, actually, because some respond, it's actually good clinical practice to treat those patients as well? And that's a really, really tough conundrum. It's a sort of clinical equipoise question. So these are some of the particular areas for me. I can't remember who said it earlier on about adverse events often being off target. That's a killer, particularly with small molecules. You don't have the same effect as much with large molecules, so with biologics, but it's one that we always have to be aware of. So anyway, I've spoken too much. I'll let somebody else say something.
Brilliant, thanks, Alex. Charlotte, are you there? You're back.
Sorry, my battery wasn't plugged in. I thought it was. I nearly lost you, but we're good. We're good. Yeah, I agree with everything Alex said. The other thing for me is when we're looking at GWAS data, it's sometimes hard to link that to an actual target, as has been mentioned, and also a function. If you take the diabetes field, for example, you often get things that are structural sort of genes, and trying to understand the biology behind why that might be important in that target is often difficult. But the other thing I think we also need is better diagnostics. If you're going to be testing these in the clinic, for coming back to what do the clinicians do, then the diagnostics need to keep up with the drug discovery angle as well. And for me, that's something that isn't just linked to pharmacogenetics. It's also, if you take the AMR space, so antimicrobial resistance, you know, we need to have the diagnostics to support the clinical developments that are coming through.
Great, thank you. Ellen, do you want to go next?
Yeah, similar to that, to what Charlotte said in terms of diagnostics. I think one of the big challenges is in using, say, data from biobanks or electronic health records is the lack of sort of standardized ways to capture phenotypes using, you know, standardized ontologies, capturing what's an adverse drug reaction versus a symptom of the patient that may be completely unrelated to their treatment response is quite difficult to pull out from that kind of data. So I think some challenges exist in utilizing, you know, we've got a vast array now of data that's potentially accessible out there through electronic medical records, but it's quite messy and noisy. Linking that to patients' treatment records and prescriptions when sometimes secondary care and primary care are not linked together is tricky to really know, okay, this was a result of this person taking this treatment in terms of trying to pull out adverse drug reactions, for example. And then, as Maria mentioned, the polypharmacy issue, if patients are on multiple drugs, which they often are, especially as we age, how do you pick out drug-drug interactions, you know, phenotypes that are responses to the combination of drugs versus a single drug, and then how do you link that back to ultimately to the targets that those drugs were hitting, to help then inform on how safe it is to modulate those targets. So those are sort of some of the challenges using big data sets, for example.
Great, thanks, Ellie. And Maria, we've already got quite a lot of challenges. Anything to add there?
Yes, as Ellen said, I think the main challenge from a more bioinformatics perspective may be sometimes the data availability, data standardization, and especially when you come to a scenario where you are not looking only at one drug, one phenotype, but many different drugs, many different phenotypes, and how do they combine?
Great, thank you. So there's lots of challenges, but maybe to sort of turn it around as to why we're doing this, which again you all alluded to. Have you got examples of where you've seen kind of things go really well, either at sort of stages of the process or even from the patient perspective? Kind of examples. I think Alex, you talked about an example, didn't you? Anyone want to share things that have worked well in this? Ellie?
I think ivacaftor and cystic fibrosis is a really nice example in this field, where we thought for a long time that gene therapy was the thing that would help treat cystic fibrosis, when actually looking at the mechanism of particular mutations in those patients has helped us understand, okay, this, for example, these are mutations that affect gating of the ion channel. Can you design a drug specifically to allow opening of that channel? And hence sort of ivacaftor and other drugs that followed on from that, you know, were designed specifically from the understanding of how those mutations affect the CFTR channel, and therefore can you design drugs that specifically target that mechanism. And so I think that's a really nice example outside of cancer, because obviously there's a lot of cancer examples that target particular mutations and mutation mechanisms. I think that's a really nice example of where it's been successful in determining particular mutation mechanisms in subsets of patients for more effective treatment.
Great. And Alex?
Yeah, that's a really good example that was just highlighted there. Thank you, Ellie. I'm going to go back into cancer, but for a different reason slightly, because this is about persistence more than anything. So KRAS mutations were known to convey very poor prognosis on patients with KRAS mutations across different indications, particularly non-small cell lung cancer, and were considered for many years to be undruggable. For some reason, we just couldn't hit the target right. We being the industry and academia in combination in particular. And over the last few years, the target has been druggable. And in 2020, the first approved medicine for KRAS mutations, sotorasib, was approved. And it really was a seminal moment. It wasn't developed by my company. I wish it had been, but you know, to see that there was persistence because it looked to be such an important driver. And I think it took 10 years at least to work out how to drug this target. But it shows that we should never give up once we find a target that seems to be of real clinical importance.
And that's a really interesting example as well, because it uses the chemistry, it targets the reactive cysteine, so it kind of uses the chemistry of the resulting mutation in order to modulate and target. So yeah.
Charlotte, have you got examples that?
I haven't got one that comes to the top of my head, but I think there has been some work in the space in the COVID-19 drugs that has been looking at this and how pharmacogenomics can affect people's responses to those kind of antivirals. I can't think of any examples off the top of my head, sorry. But yeah, I totally agree with what Ellen and Alex said. Really good examples there of how this can work. And nice to have thought of one outside the oncology space, Ellen, as well. So there are more examples, I think, in oncology than anywhere else.
Yeah. And Maria, anything to add on?
I don't think I have enough knowledge on this yet to have an answer for this, but I guess that, as Philip said, maybe cancer would be good thing to start from, especially those cancers that do have many subtypes, many genetic variability across them. Maybe it's easier to find more efficient drugs on these cases.
Great. So great to have some good examples to sort of ground us as to why it's worth looking at this area. A theme that came out again from Munir.
So sorry to cut in and so on. Maybe I can ask a broad question if I can, just taking people back a bit. So pharmacogenomics has been around for a long time, and it's largely been used for drugs which are off patent, etc., and to identify variation in genes involved in drug PK or drug PD, which then lead to changes in efficacy or safety, and that's how it's been used. But over here, obviously, and pharmacogenomics is wider than that, where it's being used for drug discovery and target validation, target identification, etc. So my question really is, and to all present on the panel, is when new drugs are being developed, there are examples of new drugs where there is information in the label which says that you should genotype for particular polymorphism, for example, eliglustat and cytochrome P452, eponymod with cytochrome P452C9, and change the dose, or don't give it to individuals with particular genotypes. So that seems to be a rarity compared to what the pharmaceutical industry is doing now, which is really using genomics to identify genomically defined drug targets. And that's a broader definition of pharmacogenomics. And just be interesting to understand, particularly from sort of how Open Targets is looking at it, and also how Spine UK is looking at it, as well as industry. Do you look at both aspects, or are you just mostly focusing on using genomics to identify drug targets? And when you do have variation in your your drug because of pharmacogenetic differences, maybe genetics, do you at the moment just ignore that? Sorry, that
I'll step in here because it's a really important question, and we always look for it. Do you need genetic phenotyping genotyping to in terms of which patients can take a medicine or not, or what the dose adjustment might be? And one of the classics over the last few years was also finding out that OATP1B1 expression appeared to be different in Japanese and East Asian subjects, and so rosuvastatin, a commonly prescribed statin, is dose adjusted based upon that. But that's a generalization rather than a genetic testing. But I can tell you that when a project comes to the interface between discovery and clinical, and there's a flag that there may be a P450 liability, it causes a heck of a panic in terms of what you do in the early stages. I mean, things like UGT expression is one that catches us sometimes as well, but we always look for it, because the risk of doing a dose escalation with a drug with a narrow therapeutic index with a potentially known predisposition in some patients to have lower clearance would be catastrophic. So yeah, it's taken very seriously, Minnie.
Charlotte, do you want to?
Oh, sorry. Go ahead, Charlotte.
That's okay. Yeah, I mean, we always consider it. I mean, we're working at the very early end of drug discovery with the UK Spine, but as much information as you can gather towards the clinic, a lot of the safety concerns are obviously around elderly patients, but the pharmacogenetics is also always considered, but probably more along the lines more strongly we consider how the genomics is going to affect the best targets. Not that we're not considering the effect of pharmacogenetics in its really real sense, but what are the best targets that are going to be the most likely to help target multimorbidities of aging, because it's that very early triage stage. But as Alex says, if something is got a known liability, then of course that's going to be considered in the safety aspects and the therapeutic potential. But the UK Spine is very far away at the moment from clinical drugs. We're very much in that sort of target ID, HIP ID kind of stages. So it's considered, but you know, it won't be as on the forefront as it will be for AstraZeneca in terms of actually, you know, making real therapeutics at this point.
And Ellie, can I bring you?
Yeah, and that's similar to open targets in that we don't look at sort of the effect of compounds. We're very much on the target identification and prioritization stage, but we can use genetic variability to help inform how safe it actually might be to modulate a target. So, for example, we bring in genetic constraint scores from NOMAD, which show, you know, over evolutionary time, how likely are you to have loss of function variation in that gene. If you have a lot of loss of function in that gene, then you can predict that it might be relatively safe to modulate that or inhibit that gene, versus if it's very constrained and you don't have a lot of mutation, maybe there's a reason for that and that it would not be very safe to inhibit that target. So we use sort of genetic information in that way to help inform, as well as bringing in safety information like pharmacovigilance data, and linking that to, you know, we know that this particular drugs that target this gene have been associated with these types of side effects, and though that might not be directly related to that target, you know, there's some information you can glean from modulating of that target may result in these types of side effects. So we use that sort of associated data to bring in and black box warnings, etc.
Right, just a call to everyone, if anyone's got any questions, either pop them in the chat or put hands up. I can't see any hands up at the moment, and I've got a colleague who's kindly helping me look out for them, but you know, we would love to hear your comments and questions. So I think all of you've mentioned uncertainty being a key thing in, you know, whether it's uncertainty in the what data should be gathered or the decision making that's going on. How do you see us, what do we need to do to help us progress despite these sort of areas of uncertainty? Is it with industry academic partnerships? Is it what would you suggest from your different perspectives? Ellie, do you want to?
I would say, coming from a sort of informatics perspective, just more data, more granular data, more openly available data, and making that open source and available to people to do research, and more structured, standardized data sets that will allow us to get more clarity. At Open Targets, we're wanting to build upon sort of the foundation by bringing in more granular data, such as single cell analysis, so that you can, in terms of sort of more stratified medicine, you get an idea of stratifying diseases, getting more clarity on which particular cell types are important in that disease, and therefore being able to prioritize particular targets related to those cell types. So I think sort of generally just more granular data will give us hopefully more clarity and more insights, and the ability to stratify, understand the disease better, which will then allow us to identify particular targets that are more likely to be efficacious in particular subsets of patients.
But and just before I come to the others, do you identify what data you want and sort of go looking for it, or how do you work with sort of identifying data that you might want to bring into your process?
In terms of sort of the platform, it's any sort of resources that are openly available that would help us inform on target disease evidence, but also building upon a sort of therapeutic hypothesis. So things like a clinical trial information that would help inform on either modulating the target, tractability of target, and safety of targets. So we sort of look out for resources that are available, and also in a format that is structured so that we can map that to standardized terms and ontologies for phenotypes, etc. But I'm very interested to know if there are data sets out there that we could bring in on pharmacogenetics or stratified medicine that would really help us inform these therapeutic hypotheses.
Brilliant. So call out to everyone on the workshop today if you've got data sets or aware of them. Charlotte, can I come to you on this question of uncertainty?
Yeah, for me, one of the big things is, in vitro and in vivo animal models and how we better equip them to replicate what we see in humans. So if you take a mouse, for example, their clearance is different. So a lot of drugs fail between preclinical and clinic because of potentially a mouse is different. So there's a lot of work with humanized models and stuff, but it's still, and Alex maybe knows more about this from AstraZeneca point of view, but it won't incorporate all the different pharmacogenetic parameters that could be out there. And the same with in vitro models. There's a lot of people working now on organoids, trying to be more physiologically relevant, but it's incorporating as much of that variation that we have in humans in terms of genetics into the models that we're testing to give the confidence that these drugs, to identify liabilities, to know, which ones are more likely to be safe or efficacious moving forward. And I think that it's a huge area, and it's not an easy thing to address, but it's needed to support the field.
Brilliant, thank you. Alex.
Sure, thank you. It's a really thought-provoking question. To go back to what Munir asked just previously, and also in line with what Ellie said, I think it's not only more access to data, but that data needs to be stratified and demographised as well. Because when we find out those risk factors for patient groups who cannot tolerate drugs as well, or who may do better on them as well, if we're not stratifying, if we're not capturing that information, nobody can put a search engine on this. Nobody can see, okay, where may these variables lie. So that's something I'd like to see. Also goes into what does it look like over the next few years, that we're better at representing diversity of patient populations in our understanding of the likelihood of efficacy, the likelihood of side effects. And I don't think we're there yet. I think that's something that needs to change. I also think this is completely subjective, but I do think we have to approach drug discovery and development with a level of humility as well, and not just I always think try and kill the drug, because if you can't, you've probably got one, rather than try and think of ways to positively reinforce what you hope will happen. So I think there's a level of sort of even-mindedness to the way that we approach targets, because it's difficult. It's really difficult. And the other thing is, it takes a lot of passion and hard work to discover and develop a medicine, to discover a target in the first place. They need to make sure that enthusiasm and diligence doesn't tip the balance into sort of over-optimistic assessment of targets or medicine. So that'll be my plea for anybody who's looking at discovering and developing medicines. Just remember all the pitfalls, and they've happened before and they can happen again. And if you've got a good one, celebrate it, and celebrate the fact that you're going to change patients' lives, because that's what we're all here for.
Brilliant, I love that. Yeah, humility is, yeah. We've had a question in the chat, and Maria, I thought I might come to you first about this, because it's talking about polypharmacy, which I know you mentioned. So thank you very much for the question, Ruby. So when having drugs to target comorbidity, and are there any current examples, is it better to have a medication to target multiple targets or a drug to target a particular gene that's causal for multiple phenotypes? Are there any examples or thinking there? And Maria, do you want to have a start at that one?
Yeah. Sorry, I am reading the question again.
So if there are any...
So, yes, so I think it is making like a comparison between polypharmacy and polypharmacology, maybe. Or identifying the main gene that I'm sorry. Okay. Yeah, so as I mentioned, I guess that I can say that maybe polypharmacology, it's another perspective we could look at pharmacogenomics from, since in these cases we only have one drug which is targeting multiple targets, and maybe so it is better to, it is easier to handle the level of adverse events and efficacy for one drug than for multiple of them, and especially when it comes to drug-drug interactions and so on. Following with the previous question, I think that something that could be interesting to do would be to have a complete picture of which is the impact of polypharmacy versus polypharmacology. Maybe a good starting point from the data perspective could be to make some kind of comprehensive comparison between current databases, like for example, open targets, which we have information for the drugs and information for the target, and compare them with other databases, like I can think about PharmJKB. So it could be interesting to start from there to really understand the level of uncertainty that we have. When you talk about uncertainty, and it comes to my mind missing data, completeness data, incompleteness data. So maybe a good starting point will be to really understand to which level we are missing data, and if the current data that we have has some bias, and so that we could invest more on generating this data and standardizing this lack of data. This makes some sense.
Great, thank you. Anyone else got thoughts on?
Yeah,
Charlotte.
right. The UK Spine has of course thought about this a lot, Ruby, and the answer is we're not 100% sure which is the best approach. You could hypothesize that there would be more safety concerns if you were targeting multiple genetic genes for the polypharmacy angle. It would be hard. Yeah. The drug discovery world, more selective is better. So if you're hitting less, you're less likely to have side effects broadly. But it may be that you require to hit more than one gene to have efficacy. So, you know, it's an open question. If anyone has any thoughts on this, I'm sure the UK Spine would be very interested to hear them. But it is something we discuss a lot, and you know, what is the best approach, I think is still to be decided.
It's a fantastic question which doesn't have a simple answer, not least because developing novel medicines in combinations, you've got to work on contributions and components. They're all very expensive to develop in the first place. It adds enormous complexity, and at the moment it's not obvious how you would do that necessarily. The closest I can think to what I call a pharmacogenetic or pharmacogenomic combinations would be in the situation where we're using targeted small molecules on drivers of oncogenes, say for example EGFR or ALK, and combining them with checkpoint inhibitors. The idea being that you kill the cells, you release a whole bunch of cellular data and antigen and fragments into the system, and then stimulate an immune response. And a checkpoint inhibitor enables T effector cells to then target what's remaining of the tumor. But it's arbitrary. That second component is based upon the likelihood that you would get an immune response to the death of the tumor cells. It's not a double targeting. It's almost an ability to facilitate the outcome of what happens in the first place.
Thanks, Alex. Munir.
Thanks. So I have two questions in terms of seeing how we can move forward. The first is, Ellie mentioned about the fact that the data in biobanks is sometimes poor in terms of the resolution of the phenotype, and it's clear the phenotypes in biobanks are quite superficial. They're not deep. You know, when I do a study, you know, we often go for a deep phenotype, really try to understand the stratification in disease, but in a biobank, it's often a very superficial level. So the first question is really, what should we be doing in the future as we develop biobanks? The UK Biobank is a fantastic resource, nobody would argue with that, but what should we be doing? And what should the pharmaceutical industry be pushing for, and academia as well, in terms of developing better biobanks with better resolution of data in there to help with the drug discovery process, but also in terms of individualization. That's number one. Number two relates to the elderly and polypharmacy and multimorbidity. So in my clinical practice, I often see patients with 6 diseases. The first patient may be different 6 diseases, second patient may be different 5 diseases, third patient may be different 4 diseases, etc., with different combinations of drugs between 5, 10, 15 drugs, and so on. So that's really complex. And if you were to do the mathematics of how you combine different diseases, you get an immeasurable number of combinations. And so really my question is that we do not know enough about the mechanisms of multimorbidity and so on. So at the moment, developing drugs is actually impossible to be able to treat multimorbidity. And we need better resources to be able to do that. So that's the question. That's the second question. What can we do to be able to understand multimorbidity much better in order to move it forward?
Brilliant. Thank you, Munir. So let's talk about the biobanks of the future first, and then come to the multimorbidity. Does anyone on the panel want to start on, maybe Ellie, do you want to start on the biobanks of the future? What should we be doing?
Yeah, I think it's probably more the clinical electronic medical records that are being able to link those to biobank data more easily. I think capturing longitudinal data across patients' lifetime will really help us know when particular, say, symptoms or signs have started. Was that pre or post a drug treatment? So I think, and then just finer phenotyping, which is very challenging, either for those collecting that data for biobanks, or, you know, a clinician in the clinic who only has 10 minutes, for example, with a patient. I think one of the issues, say, with electronic medical records is that individual may be seen by different clinicians, different nurses, etc., and they will be entering the data slightly differently and capturing maybe the same response. We've shown previously, actually, that the same sort of adverse drug reaction might have multiple different names, you know, and so it's captured differently in different areas of the country, for example, in the UK, through the NHS systems. So I think sort of standardization of how those phenotypes are captured. It could also be, another way could actually be allowing participants to add their own phenotypes and be able to submit their own data, you know, oh, actually, I do have a headache on day 3 of this drug that I've taken, which you wouldn't normally report to the doctor. But maybe that could be another way of gathering more granular data from the actual individuals themselves on how they're feeling.
Great, thanks. Charlotte? Biobanks of the future.
I wasn't going to comment on that one. I was going to take the multimorbidities.
Okay. I'll come back. Okay, Alex or Maria, anything on else on biobanks of the future? Okay, Maria, did you?
I agree with Ellen. We have talked about this a lot, so kind of have the same opinion on this.
And all I can say on this one is, when I see presentations coming through into the clinical space, every company I've worked at over the years, people tend to talk about deep phenotyping. in terms of their discovery programs. So if that's a commitment there, then that's good to hear. But that's proprietary information. That's not necessarily out there in the public domain helping everybody else. So maybe that's something that we need to look at. Where are the non-competitive sharing spaces that will help everybody?
Great, Sheuli.
Can I just come back on the biobank issue before you go on to the next one? So yeah, if you ask a busy nurse or doctor to be able to record data in depth, they've got a very short time. It's not going to happen, unfortunately. And often, if you look at, let's say, a skin rash, often it's recorded as a skin rash in the notes. It won't be recorded in any greater depth and so on. So we do need to think about data standardization, having people who actually do the data standardization at source and putting that data into databases and biobanks and so on. And that's going to cost a lot of money. But I would also agree with what Ellie said in terms of actually allowing participants to upload their own data, having an app which allows them to actually upload their own data. We've learned a lot from patients, asking patients what kind of rash they had or what kind of adverse effect they had. And really that is going to be important, allowing them to upload their own data. And, obviously some of it may be subjective, but actually it is important to have a look at all the data that patients want to provide. And I think that's going to be important in empowering people as part of the biobank going forward, because we're never going to have enough capacity, enough resources to allow clinicians and people at source who are recruiting for biobanks to put all that data in. And it's not recorded very well in medical records. Clearly, we could go towards standardization of those medical records. That's going to take a long time. But in the short time that we have, we need to be able to think of other ways, more novel ways of being able to gathering that data.
And I think informatics tools are really vital there, making it as easy as possible, doing all the standard, automatic standardization for you. And there are techniques out there, for example, NLP that can go through medical written notes, and pull out key data from that. So informatics could help there.
Great. Okay. Alex, did you want to come in on this?
Yeah, just a couple of points there, particularly that Munir raised, made me think a bit more. So we create apps that make sure patients take their medicines, particularly in patients maybe with neurological conditions. So it should be possible to turn those apps around. And it's also I just wanted to highlight, because this is something that's hitting us in terms of questions about Project Optimus with FDA at the moment and optimizing doses, that Friends of Cancer Research and FDA are working quite heavily on the concepts of better patient-reported outcomes, and I think that's going to come down the line as well. So I know a number of companies are working with FDA on this side, so hopefully that will improve over the years.
Great, thank you. Okay, let's go to the next question, which was about multimorbidity and combinations of diseases, and sort of thoughts on how we tackle that. And Charlotte, I'll come to you first on this one, if I may.
Thank you. Yeah, I mean, Munir, we don't know enough is the answer. But the way the UK Spine has approached this is to, rather than look at the disease itself, is try and look at common pathways that underlie those diseases and of aging. So things like senescence, inflammation, and then if you've got targets in there that hit multiple pathways, they're more likely to have efficacy in multimorbidities. So the answer is we need to understand more about multimorbidities for sure. We need more research. We need more people looking into this. And I think it is an area that's picking up quite quickly across the research fields. But, trying to pick out common things between those multimorbidities is how we have approached it at the UK Spine. And I think also we need to, a lot of it's about changing thought process, I guess, with clinicians as well, because at the minute, as you say, people present in different ways, that they'll have one disease first followed by another, and your next patient may be completely different. And it's gathering enough information about how people age in terms of their multimorbidities and what predisposes people to those multimorbidities as well in terms of aging that can help both clinicians and drug discoverers, I think, identify the best approaches to put people on less drugs. You know, no one wants to be taking 20 drugs of which no one really knows what the interactions between those drugs are. And I think that highlights another area that needs to be researched is with common combinations of drugs, you know, do they actually work together or not? And should we be prescribing in a different way is another area as well that I think we need to be looking at.
Great, thank you. Anyone else want to come in on thoughts on multimorbidity? Ellie?
Yeah.
I'll let Alex go first.
Please, Ellie, go ahead.
Oh, I was just agreeing with Charlotte, and actually at Open Targets, we've had a project on sort of network analyses to try and pull out those common pathways, or looking at the pleiotropic effects of targets, which could then, you know, work to, if you modulated that target, it could hit multiple different diseases or phenotypes. But then the risk is then adverse reactions sort of beyond those networks. And we can look at genetics as well to see if there are targets, for example, in aging, where you've got signals for the same target for multiple diseases, to see if that would be a good target for multiple conditions. But yeah, a huge issue in polypharmacy and multimorbidities is trying to pull out from that data what are the true signals and what are drug-drug interactions, and it's a challenge. I'll leave it over to Alex.
Alex?
Sure. Well, it's a question of panacea, almost here. And I think all we have is, through close clinical pharmacology in particular, a diligent work is to explain whether you can co-administer drugs together or not, rather than why, quite often. And I don't know how long that would take to get to the latter. But I suppose there's also a question of what's linked in that polypharmacy. Which of the diseases are linked together versus are they completely disparate from each other or separate from each other? And I don't have a clever answer for that, Munir.
Do you think that polypharmacy sometimes exacerbates the disease multimorbidity?
Very possibly. Yeah. It's effectively iatrogenesis, isn't it? The process of trying to make something better, you make something else worse.
Yeah. Maria, any thoughts on this one before we?
Yes, I just wanted to mention that in Open Targets, we have built as an example to how to contribute understanding how these different targets are implicated in a disease. We have built a web application that is called Target Age, which is trying to identify which are the causal genes that are shared between pairs of diseases that are associated to aging. So the paper is about to be published, and maybe it could be interesting to explore it. Just as a case example.
Great, thank you for sharing that. Okay, we've got a couple of questions in the chat. So the first one, back to level of data. So will our future health provide the level of phenotypic data that's currently missing? Anyone feel able to tackle that question? Munir?
Yeah, so I can obviously open Our Future Health is a great initiative to recruit 5 million individuals. Clearly, the sort of challenge is to recruit the 5 million individuals, and then the second challenge is to actually follow them through longitudinally, and the ability to do that will be limited given the numbers that have to be looked at. Clearly, there will be more granular data in certain disease subtypes where there will be more close follow-up and so on. But overall, in terms of long-term follow-up and what will happen and how good that data will be, we need to see in terms of resources available, but also in terms of how the participants will actually interact with Our Future Health in terms of providing their own data and their own, participant reported outcome measures and so on. So I think it is too early to say exactly what the level of phenotypic data will be. In some, it'll be very good. In the whole 5 million, difficult to say at the moment until we see how it works.
Great, thank you for that. Before I go to the next question, there was a hand that was going up. Was it Imran? Did you want to say something, Imran? Or
no, I mean, I was just agreeing with the previous conversation about data quality that Munir and also Ellen have picked up on. So I'm a GP working in primary care. We have sort of 2 or 3 main IT supports that support the electronic health record. The level of recording, the quality of recording is obviously an important issue. We have multiple ways in which patients can now access GP surgeries. And so when you talk about uploading data from patients, the challenge really is to have a standardized way in terms of data extraction and how that data is recorded on the record. But yeah, no, very interesting discussions. Thank you.
thank you, Imran, for that. That's helpful. Right, back to the chat. We had a question about prescribing cascades. So prescribing cascades in primary care are all too familiar, and polypharmacy is a real issue, especially in older patients. I feel clinicians need a greater exposure to clinical pharmacology going forward. Would anyone like to come in on this? Charlotte, is that?
I mean, I'm not a clinician, but I take the point. I think, you know, the more we can equip our primary carers with information in terms of these things, then the better. You know, yeah, I don't know if Munir has more more insights into this area than.
Yeah, maybe I can add something to that. So I completely agree with that. But I think if you consider that the current standard for complex cases such as malignancy, etc., and you know, even if you have a bad fracture, you have a multidisciplinary team who then decides what to do in terms of, you know, what kind of surgery you need or what kind of targeted therapy you need, and so on. But actually, we've got an aging population with polypharmacy, multimorbidity, yet it's often left to individuals, GPs, to be able to sort it out, or pharmacists do the medication reviews. And GPs are great, pharmacists are great, you know, but not, you know, nobody has enough knowledge to be able to look after those particularly complex patients. And what we need are really for the future are multidisciplinary teams, which includes GPs, pharmacists, clinical pharmacologists, disease specialists, etc., working together to really understand what's going on with the elderly with polypharmacy, multimorbidity. Because we're not going to get, unfortunately, we're not going to get the sort of magic bullets to treat multimorbidity in the near future. Sorry, Charlotte. I mean, I don't think we are.
No,
but what we need
I agree. We're far away from it.
Yeah, and so what we need to do is to look at what we do at the moment with our patients, and really understand the pharmacology, the disease interactions, the drug-drug interactions, disease-drug interactions, disease genetics interactions, and that requires a multidisciplinary team to sort that out. Now, clearly, you may say that's going to be impossible to do because of the resources required and time of people and so on, but we need to think of a different model of care than we have at the moment.
So there are companies at the moment that are using algorithms to help with optimization of dosing based upon patient demographics, predisposition, etc. I would love to see in the future some form of neural network where a physician can type in patient on XYZ drugs. What are the risks? What are the potential dose adjustments that you may need to be able to take? That would just be super cool. That would help in terms, and it would capture as well, rather than the knowledge staying with one person. It would be there as a repository for everybody to take in, and that's going to require a huge multidisciplinary effort with funding. But wouldn't it be great?
Yeah, and I often start off with a slide like that. This is the future prescribing, where you have AI models, pharmacometric models, together with all the different parameters, which allows you to be able to define the dose and drug. But we're a long way away from that, Alex.
I think so.
Brilliant. Okay, Imran, did you want to come in on that?
No, I mean, I completely agree. And we are working closely with our pharmacy colleagues in GP practices now, and structured medication reviews and so on are beginning to be the norm, really. I'm just thinking about epidemiology, really, and how these diseases over time are linked. And I'm not sure Charlotte has got the answer to this, but there's good epidemiological data around multimorbidity and the type of diseases that can be linked together that come up commonly. And maybe it's a question of anticipation and yeah. What do you think, Charlotte?
Yeah, I think epidemiology data can be very powerful here in terms of predicting what diseases could go together. I'm not sure it always pans out in every patient, of course, but it's informative. And the way we've been approaching things in the spine is to try and take all these different data sets and epidemiological data to try and inform how we approach trying to target multimorbidities of aging. What we need is some tool molecules that we can test in some models and prove that the concept works. That's where we're at. Yeah, and the idea is that you try and delay the time that the second morbidity occurs. So you try and have people healthier for longer before they get multimorbidities, essentially. And again, we need time to prove that concept can work, and we're not there yet.
Great, thank you. Okay, we're coming up to the end of this session, so I'd just like to kind of go to each of our panel who've been absolutely brilliant and just ask about your thoughts. So, 10 years from now, where do you think we'll be? I think Alex has already started to describe what he'd like to see. But yeah, if you could just sort of share where do you think we'll be, or where would you like us to be in sort of 10 years from now? Ellie, can I come to you first?
Yeah, I think utilising all the useful data that we have to be able to predict, okay, for this particular patient, be able to simulate and predict, well, better characterise their own disease and the interaction between their germline and potentially the somatic genome and how that's affecting their disease, and be able to predict from that what target would be best in that patient, and therefore what particular drug or modulation for that patient, taking into compass all of their symptoms, their genetics, so the multi-omics, and be able to predict that for them. That's probably beyond 10 years time, but more personalised medicine for that individual. And as was alluded to, maybe also predicting their risk of particular diseases in the future, or developing a comorbidity, for example, and how can we prevent that from happening before it does. That would be the ideal.
Charlotte?
Sorry, I think it cut out there. I think it was me. For me, I'd really like to see standardized in vitro and in vivo models that incorporate known pharmacogenetic liabilities, things like that, across the board. And Alex's point about having some kind of access for clinicians to be able to easily, in terms of multimorbidities and polypharmacy, look at these things and see what they should be prescribing or changes, it would be phenomenal. Whether that could be done in 10 years, I don't know. It's a big thing to have to produce, but it would be excellent. It really would.
Brilliant. Maria? Oh, we can't hear you. You mute.
Yeah, sorry. Maybe to be more conservative, I would like to have this data more standardized to be able to include it in platforms like Open Targets, which I think it's a wonderful tool that we could explore more and more. And so incorporate this data to allow us to take into consideration pharmacogenetics for the very beginning of the drug discovery and target identification process. and from there to the sky.
Brilliant. And Alex?
It's a bit like writing a wish list for birthday presents, really. But I'd like to see big data be meaningful, relevant, and accessible. I'd like to see a better understanding of things like immunological differences between non-clinical models and patients, because I often think that's a pitfall. We assume that the immune systems are more similar than they are. I would really like to see improved access to global data in terms of drug interaction liabilities, patient demographics, etc. But I'd also think about not just better information, a better understanding of what to do in terms of treatment of patients, but actually a better understanding of what not to do. When you consider the position paper that was shared, that over £500 million a year spent on dealing with adverse drug reactions. Now, we could eliminate that or greatly reduce that cost and patient cost could be redirected elsewhere. So we never lose sight of that. Just avoiding doing bad things is it's often just as important as doing the good things in the first place.
Great. Thank you so much to our panel. We've had a fabulous conversation, and thanks to everyone for your questions and comments. And I'm sure that there are lots more thoughts and questions, so please do bring them into the workshop groups that will be after the break, because we want to make sure we have a really rich conversation there. We now have 15 minutes to have a break, and then it's back here into the Zoom at 11 o'clock, where we'll get the introduction for the next part of the workshop. So lastly, just massive thank you again to the panel. Really appreciate it. Thank you.
Thank you.
So the idea now is just to ask for some feedback from each individual group and any questions or final remarks you have before we close the workshop. So really the first group, which, let me just get my papers. First group, which was how is pharmacogenomics currently used throughout the drug discovery pipeline. So Harriet was chair and Claire Denton was rapporteur. So I don't know who's going to report back to the whole group.
Claire, would you like me to? And then you can shout if I've missed anything. Okay, so we had a great conversation that covered quite a lot of ground. So I'll try and do it justice by covering the highlights. But the question of how pharmacogenomics is currently used throughout the drug discovery pipeline, we had quite a range of experience and perspectives within our group. And we started by talking about industry interest and touched on the idea that pharmacodynamics is perhaps not quite as developed within an industry perspective as pharmacokinetics. And this moved on to discussion around the variation in clients interested in this space, and some companies that are very specifically using genomic targets and some that don't yet. And this led us into a really interesting conversation around our disease definition. And one of the opportunities, but also the challenges in this space, is that the definition of disease is getting more granular as we have more information and can split them up. Breast cancer was an example here. And this then, I think one of the key things that came through our conversations was around the role of patients and the challenge with sort of explaining some of these complex ideas to patients so that it actually usefully feeds into how they are managing their own condition and understanding their care. The opportunities for a learning health system approach and the data that could be available were discussed, and we talked about the sort of how we get the most out of that data and where we're gathering data, particularly, potentially we've got samples that could provide genetic information but aren't necessarily being analysed at this point, and how we kind of preempt what might be useful in the future versus balancing what is appropriate to gather and use. We then looked at the sort of information for patients and what is allowed. The Netherlands were put forth as an example of where they may be a bit better at how they pitch their information to patients. And then we looked at sort of this interface between the clinic and research and how data are being pulled across. So I guess within the pipeline, we then got quite focused at the clinical trials end of things and what that then means for patients understanding their care. And we finished on some really interesting discussion around stratification of patients at a trial phase, such that actually they're kind of representative and transferable into the general population who are then using those drugs, and how we have data feedback between those two things, because it really is such a complex picture for patients, and making sure the target population is representative is a real challenge. So we had lots of discussion. I don't know if anyone wanted to throw in anything I've missed that was particularly burning.
Claire Denton, do you want to add anything as rapporteur? You're muted.
Doesn't look as if she's able to take it off. No, it's
Can't
not working.
Okay. Okay, any comments or questions? So Harriet, I don't know whether you discussed within your group about, when you were talking about patient information for patients, what patients expect and what patients understand about genomics and the limitations of it, because genomics is not going to be the answer for everything, and maybe only be partly the answer for some things.
Yes, so we certainly did have a discussion on managing the complexity of the topic versus what is actually useful when making a decision as a patient. And there are certain conditions where patients are perhaps required to sort of familiarise themselves with that information more by virtue of sort of what they don't know and what is needed to actually usefully treat them. And I think rare diseases is probably a good example of that. But I think there's quite a lot more that could be done, particularly where polypharmacy is involved, to actually think about the sort of whole reality of the patient and their experience, and then what is genuinely useful within them. And actually, one point that I didn't mention was around prescribers trying things and then stopping them if they don't work. And we had a bit of a discussion around whether prescribers are becoming more risk averse because of the additional data that they actually perhaps have at their fingertips with pharmacogenomics, and what that means in terms of sort of where there's a grey area in terms of stratification of patients. There's quite a few patients that will sit in that grey area where it might be useful, but it might not be, and we don't really know how to manage that. So I think that was a really, really interesting point that came up.
Okay, and I just wondered whether, how many companies, I know that some big companies have published on this, such as AZ, GSK, etc. How many companies are now using genomics data to identify drug targets and so on? Is it now widespread? Is every drug company using this? At least partly, or sort of. I don't know whether that may not have been discussed, but does anybody know the answer to that?
I put it to Alex, I think.
I think pretty much all. There was a big drive a few years ago about, one of the things that I think some companies had slipped into was using non-clinical models that could be tractable chemically, rather than feeding back information from clinical situation. And they moved away from that because failure rates got very high in terms of successful medications. So I'd be very surprised if there were any companies who didn't incorporate these approaches.
I would support that, because and the fact that all the knockout models and things have become so much more available to the smaller companies, it means that it's much easier to start identifying things earlier on. In terms of pharmacodynamics, and I do think that it used to be really hard to find in vivo models that would help you once you targeted something. It's much easier now. My mailbox is full of new models that keep popping in from various places saying, send your clients to us.
Okay, great. Thank you very much. Is there anything else you want to say, Harriet, before we move on to the next group? Yeah, okay, that's great. Thank you very much for that. So that was group one. Group two was how could pharmacogenomics be used to inform drug target identification, stroke prioritization, and Ellie McDonagh was the chair, and then Lisa was the rapporteur. So I'm not sure who's going to report back.
I think we've got maybe Phipps now, yeah.
Oh,
So it's me, and I actually have to cover a very broad range of topics. So we started by simply looking who is actually using it, and it quickly turned out that okay, we have this effort from GVAS to identify new targets, but actually I think it was raised that most successful drug targets came from like rare loss of function studies. So this was what is now available with whole exome data in UK Biobank, and possibly all of us and all these large resources might be most beneficial in terms of identifying good targets, because there's still this challenge if you have a common variant which sits somewhere in the genome, possibly intergenic. How can we link this to a target? And then the conversation got easily onwards to, even if we have a target, how do we establish where it is actually expressed and at which levels, and what is the difference between establishing this on a transcript based view versus do we have evidence that the protein actually as a primary target of small molecules and so on is expressed to the same extent. And this was more or less on the target identification, but then a huge interest is obviously on how pharmacogenomics informs patient treatment for the one sitting in front of you. So how do we get to the point where we can personalize, and how can we integrate other data layers like imaging and so on to get closer to stratified populations, always working around this like trade-off between having the sufficient sample size to see genetic effects compared to like very stratified phenotypes which may only cover fairly small sample sizes. So I think that's why I said it was a very broad discussion. It was quite interesting to see. And then also, even if we have a target, we have an idea what might be there, that we see discrepancies in the data where transcript expression would say, okay, an antagonist is needed, but protein might say an agonist might be needed because you see different effect directions. How can we start resolving those? And then diving deeper into some like particular characteristics of the data we are looking at. Proteins measured in blood, whereas transcript can now be measured in tissues, but even in single cells. So single cell is obviously one of the emerging technologies which can link better to genetic data to get really to because we're not treating a tissue, we're treating hopefully the cell type which is responsible for the disease phenotype, right? So how can we incorporate those? And I think this is also an open question from open targets to the community. What are resources to be integrated? Single cell data might be one of those, but also at the end quite rightly raised the influence of the microbiome. So how do our gut microbes contribute to what is actually available in the circulation in terms of modified substances which may or may not exist, or whether they contribute to catabolizing effects and these kinds of things.
Okay, that's great. Thank you.
That's a lot. That's why I said it's quite diverse.
Okay, Ellie, do you want to add anything?
That's really well covered. Thank you, Mark. Yeah, it was very diverse discussion and raising lots of questions, but looks interesting discussion.
Okay.
Thanks.
So Mike said about microbiome and so on. At the moment, Open Targets doesn't incorporate microbiome data in your databases. Is there the plans to, and how are you going to standardize that?
Good question. I think at the moment, as we discussed in the group, it's not quite clear yet what the effects are, both of the microbiome on drug response, but also the other way around. So drug response, drugs on the microbiome. So I think once there's a bit more, I guess, once the evidence builds up, we can start to think about how that could then help inform maybe targets that are not so much influenced by the microbiome, maybe on would probably be how we'd maybe use it.
Okay. And question to anybody really is you know, and I don't know whether this was discussed in the group, is that if you look at most of the GWAS loci which have been identified for complex disease, the majority are intronic, you know, with the functions not known, but we're learning more and more about enhancers, super enhancers, etc., and so on. So how do we utilize that information to start thinking about drug target identification, prioritization? I don't know whether that was discussed, Ellie.
So sort of variants that are identified through GWAS that are non-coding. Yeah, so we're actually, well, at Open Targets, we bring in, we have a machine learning pipeline that helps us predict from GWAS what's the most likely causative underlying genes for those and prioritize those. So we bring in both the GWAS and functional data like PQTLs, EQTLs, but interestingly, it was raised in our group on what happens when you see the differences between the opposing effects of an EQTL and a PQTL, which causes challenges. But that's how we handle it in Open Targets for the genetics portal.
Okay. Any other comments or questions for this group? If not, thank you very much. So the third group was on personalized prescribing, and Emma McGovern was the chair and rapporteur was Hannah. So I'm not sure who's reporting from your group.
Hannah, would you like to, or would you like me to?
I don't mind, to be honest. I mean, I can do a quick summary and we can tag team. So yeah, the third group was on kind of the opportunities for drug discovery that can be facilitated by clinical implementation of pharmacogenomics, and really like bringing this into patient care is not something that's been done before on a national level. So the sort of starting point was the BPS multi-stakeholder report looking at how to best move forward with this. And so the kind of underpinning question was, how do we optimize the value of clinical data for R&D? And we talked a lot about the real importance of engaging patients on this and building public trust around the topic, and how to really incentivize patients to give their data to this type of research, with patients becoming kind of stakeholders in their own medicine generation. We talked about how it would be really important to make sure there were concrete examples of showing the efficacy, the real benefits that can be brought by pharmacogenomics being implemented in patient care as a real way of showing why this is important. We touched a bit on the kind of data being collected, the quality of that and the depth of that, which we talked about a bit in the first session, but that was raised again as something that's really important, and integrating data across, integrating pharmacogenomics across with other data sets. The importance of cross-sector collaboration on this, and the kind of ultimate goal of making sure that there's, well, one participant described it as a kind of virtuous circle of patients benefiting in care, and then that information feeding back into the drug discovery process. Emma, I don't know if you want to add any other thoughts. I know there were loads of really good points, and I'm sure I've not captured all of them.
No, yeah, you did an amazing summary of a complex and very interesting discussion. Thanks so much. I think we, after touching on all those points, we're a little bit just overwhelmed by the size of the question, and the difficulties in dealing with factors like gene-environment interactions that may be poorly characterized, and issues like phenoconversion as well, and that maybe, you know, all of that needs to be taken into account in the first instance. But for those of you who know me, I did not lead on this at all, but the importance of public trust and patient engagements.
Okay, any other comments or questions on that particular group? So just coming back to the patient engagement aspects, how much and I think, Emma, you've done some work on this and maybe read about this and so on. How much knowledge out there is there in terms of the public, in terms of genomics and drug target identification, but also in terms of personalization of dose and choice of drug and so on? And what do we need to do to be able to improve that?
Thanks so much. I'm working on a project now to try to do some in-depth focus groups and surveys of the genes and health population, which is a London and Bradford, Manchester-based cohort of British participants of South Asian background, South Asian ancestry, who donated their DNA for a biobank type resource. And I'm going to try to work with them to understand this a bit better, because in the literature, there's just not that much. And a lot of the thematic reviews which are in the literature, either focus groups or surveys, are actually gathering a lot more the perspective of the prescriber. In terms of the participant perspective, there is some very, very high level sort of general feedback, but it tends to be sort of small groups of people, and it tends to be the people who engage more in genetic research in the first place. And it doesn't tend to answer the questions that we have about clinical implementation. The really broad themes that emerge from those is that, as exactly our group has said, and you've said in the past, that trust is incredibly important, and that transparency and dialogue and engagement really, really need to happen. And that the participants who had feedback after the fact from biobanks of pharmacogenomics reports would have liked to have been asked before what information they wanted, so that patient input in terms of what information people want and how to provide it to them.
Great. Thanks very much, Emma. Any other comments anybody wants to make on any aspect of the workshop we discussed today before I close the workshop? Okay, not seeing any hands up. So I think we're nearly at the end of the workshop, which is due to finish at 12:30. So first of all, to say thank you very much for everybody who's joined us today from many different countries. It's great to see so many people, and it's great to see, hear the discussions in the morning, as well as in the breakout groups. What I'm hearing, and correct me if I'm wrong, if you sort of disagree with me, is that we are at the start of the journey. Actually, there's an awful lot to do. There's an awful lot we don't understand, but we've got to start somewhere. And we have resources, but the resources need to be improved. The resources need to become much deeper in terms of what we have. And I think that although pharma may be working on this, I think it's also important to make sure there's public-private partnerships in order to move this forward, because it's too big a space for one particular sector to be able to achieve what we need to be able to achieve in terms of trying to improve the health of our population. I think in one of the working groups I was at, Flick said that we need to move away from the sickness service that we are to the preventive service that we need to be. And that is going to take a lot of doing. And particularly with our aging population and so on, to make sure that we can, our aging population can remain as healthy as possible for as long as possible. The health span needs to increase, and that will require a very much preventive approach, understanding genomics and understanding how we can actually intervene early on. And the work that's going on in multimorbidity is fascinating, but actually we are just at the tip of the iceberg in terms of what we need to understand and know in order to develop the drugs and drug targets, identify drug targets for future treatment of those particular comorbid conditions. So I think there's a lot of different things which have come out from here, which I've really enjoyed and sort of learned. So it just leaves me to thank everybody, thank the chair for this morning, Sheuli, and the panelists for this morning for their discussion, and then for the chairs for this afternoon, for the breakout groups, as well as rapporteurs, as well as all the participants. Thank you very much for your input today. I hope you have enjoyed it. Please let us know at the UK Pharmacogenetics Stratified Medicine Network what we could have done better today, or whether you enjoyed it or not. Any feedback is important for us to be able to take forward into our future workshops to make sure that if we haven't got it right this time, that we get it right next time. So thank you very much for your input today. And it's great to see everybody, and lunchtime really. Thank you very much, everybody.